Demosponges are the most diverse, abundant, and in some ways the most spectacularly derived of all living groups of sponges, and they've long been used as a model for the earliest animals. So their fossil record is actually very patchy, but in this video we're going to trace it back as far as we possibly can and see where exactly we end up. Welcome everyone, or welcome back. So today I'm going back to a series that I'm doing on the evolution of sponges. This is the area I know best. It's where I spent most of my research career and, well, career in a sense. I've published dozens of papers on this, and it's where I have the most opinions. It doesn't mean necessarily that I have everything right, because that's science. You build up ideas based on the evidence, and sometimes those ideas get proved wrong. And that's actually how science progresses. But I'm going to present to you how I see the history of demosponges, and we'll see how convincing it is. But this is our current understanding of a fairly large field in paleontology. In this case, there are multiple lines of evidence with different levels of reliability. Sometimes it can be difficult to tell how you should weigh molecular clocks against fossils, against biomarkers, against all sorts of other possibilities. And this is going to be an attempt to show you all of the different angles and balance how convincing they are, how well they fit in with our other lines of evidence, and see where it all leads us. So, demosponges. They include about 5,000 living species, by far the most diverse group. In fact, it's about 85% of living sponges. They include the only freshwater sponges. They include the only predatory sponges that catch fish and eat them, in a rather grim and grisly way, but that's probably one for another time. They also include reef-forming rock-like skeletons that are quite major contributors to the fossil record of reefs. They include the bath sponges, which have been harvested by humans, and indeed by dolphins too, for thousands of years. They're an extraordinarily diverse group, and they have colonised everywhere from mountain ponds to the deepest oceans, and the intertidal zone in abundance. As a result of all this, they are by far the most familiar sponges to zoologists and the general public alike, because they're just found in the most easy to find places. And as a result, they have been considered quite often to be a sort of archetypal sponge. They're often considered the basic sponge form, if you like. The problem is that these sponges are inhabiting an extremely difficult environment. So they've ended up adapting to an extremely turbulent environment, with rocks being thrown at them by the waves all the time, vast changes in salinity and temperature. It's a very hard place to make a living, and for that reason, these very shallow water ones, and freshwater ones as well, end up being completely different to a lot of their cousins. So what we see is a whole array of compound, colonial, encrusting, amorphous things with no particular shape. They just adapt themselves to whatever the surface is. They're packed full of tiny little spicules of opal, which are a deterrent to anything trying to eat them. They're also packed full of noxious chemicals in many cases, and that's only one facet of the extraordinary diversity of this group. So we're going to need a starting point. We're going to have a bit of a census of the modern diversity of demosponges and try to peel it back to what was the ancestral form. The details are still being sorted out, it has to be said. Demosponges are an absolute nightmare for traditional taxonomy, because they have so many features that are not just evolutionarily plastic, but actually plastic within an individual, that they depend on the environment. The amount of silica dissolved in the water can change how much they mineralize their spicules. It's endless. And a new order of demosponges was actually defined in the last few weeks. So it is an ongoing process, but we are getting closer and closer to at least the basic pattern. So the fundamental divisions look like this. we have a split around the base into two main divisions. One of these is the Heteroscleromorpha. These are the ones that have abundant spicules. Most things that you might encounter washed up on the seashore are probably heteroscleromorphs, although that doesn't quite hold because that's one of the other ones. We'll come back to heteroscleromorphs in a moment. The other group is a little more complex. It is composed of the Virungiomorpha and the Keratosa. The Keratosa are the most familiar of all the sponges, really, because they're the ones you can buy in the shops. They've been harvested for a long time, thousands of years. The ancient Greeks had quite a big industry in these. And they have a purely organic skeleton of spongin, almost entirely spongin, which is basically a collagen-based substance with quite a lot of sulfur in it. But it's very tough, it's soft, and it gives them a skeleton which doesn't have any spicules in. On the other hand, you also have the Virungida and the Chondrosida. The Virungida basically look largely like keratosans, but their skeleton is made mostly of chitin rather than spongin. And so it's a polysaccharide rather than a protein. It's a different substance. But it has a very similar fibre network skeleton. Strangely though, they also have calcite and or silica in these fibres. So they are, in some cases, complexes of chitin with thin layers of aragonite, calcium carbonate, and opal within these fibres. So they've been reinforced with minerals. The chondrosids, in contrast, generally have a skeleton of spongin fibres. But the chondrosids and the virungids are sister groups, but here we have spongin, here we have chitin. Except that some chondrosids don't actually have a fibre skeleton at all. Some of them do, but also have spicules. Asterose spicules, spherasters, which are odd little things, basically tiny spiky balls. And these are the only spicules within this entire half of demosponge evolution. There is also a keratosan called Ernstilla, which has a fibre skeleton which is made at least partly of chitin. So although we can summarise the features of each of these major groups, the divisions are complex and they break down when you look in detail. There's always an exception to everything that we try to generalise. When we're talking about the chondrosids that don't have any fibre skeleton at all or spicules, they're basically just a mass of gelatinous material but with quite a lot of disaggregated little fibrils of collagen sort of embedded within the tissue to give it a little bit of structure. But even so, they're encrusting amorphous things onto hard surfaces. Okay, so that gives us a basic pattern for these things, but the question then is, what was the ancestral condition? About the only things we can be certain of is that these non-spiculated demosponges, in general, probably had a combination of spongin and chitinous fibres, or one or the other, and in one group there are some unusual spicules. So ancestrally we can assume that they had a fibrous skeleton made of some combination of spongin and or chitin, possibly with spicules that have been lost in most lineages. That's just one half of modern demosponges, of course. Within the Heteroscleromorpha, which is everything else, there are spicules everywhere. Well, there's the odd exception, like the rather fabulous cave-dwelling Myceliospongia, which is convergent on a predatory fungus. Sponges are weird. Anyway, that said, aside from those exceptions, the Heteroscleromorphs are entirely spiculate, and they are extremely diverse. So this includes the great majority of living demosponges, all 5,000 of them. So there are currently, I think, 17 or is it 18 now, orders of Heteroscleromorph demosponges. And these are very hard to define quite often. They've only been sorted out with genetics. People have tried over the last century or two to try to classify them based on the skeletal arrangement, the spicules and so on, and have led to wildly different classification schemes, which have now been superseded entirely by genetics, which seems to be the only way to really sort them out. There's one order which is distinctive on the base of the skeleton, totally distinctive, which is the Tetractinellida. And these, as the name might suggest, have tetractins. So they have little four-rayed spicules where the rays are all at sort of odd angles to each other, like a caltrops. So that is one order, and that seems to be an innovation of that order, because we don't really see those spicules at all outside of the Tetractinellida. Psst, you also get them in the Calcarea and the Homoscleromorphs, but don't tell him. The rest of them all have monaxons of various types. There are some which have a rounded end, some with a little ball on the end of it, but mostly they're tiny little oxias, which are basically just tiny needles pointed at both ends. And they can be arranged in all sorts of different ways, little tufts and networks bound together, combined with an organic fibre skeleton of spongin. The proportions of the fibre network and the spicules varies enormously throughout the group, but in general what you have is a fibre network skeleton, like in the keratosans for example, but within that are embedded a substantial number of spicules. So the spicules themselves are very small and simple, they can't interlock. So what you have is a way of actually forming a rigid structure with them. It's a combined approach which works extremely well. They're still fairly tough, they have a little bit of give to them. If you throw a pebble at a Hexactinellid, it's going to break. If you throw one at a demosponge, it will probably squish a bit and bounce off. And the demosponge won't care anyway because it's a compound thing and it can lose half of itself without noticing. I should also say at this point that this is the group which has, on numerous occasions it appears, evolved a massive skeleton. So the lithistids, the name means rock-like, have dispensed with the organic skeleton almost entirely, and what they have instead are overgrowths of the spicules, so they're no longer little needles, they've actually got sort of weird club-shaped and convoluted interlocking ends, so the spicules then grow together to form a structure that is extremely tightly bound together. And these things can form reefs, they have done in the past. In the Ordovician, the early part of the Ordovician, the dominant reef-building organism was lithistid sponges. Then there are others that haven't modified the spicules, or they have in a way of losing them, but instead they have hypercalcified. They produced a coral-like massive skeleton of calcium carbonate, aragonite or calcite, and this has sort of overgrown the original skeleton that was there and replaced it. So you get groups like the stromatoporoids, probably the archaeocyathans, the catetids. Today there are very few examples of the sclerosponges, but in the past there have been several groups that have repeatedly created this hypercalcified skeleton, and in many cases we're not even totally sure what they evolved from. They've lost the original skeleton so completely that there's little trace left. But in at least quite a few of them, we're pretty sure they were demosponges, and heteroscleromorph demosponges of some sort. It's a real shame that these hypercalcified ones are actually of little use for studying the evolution of demosponges, because they are all very derived, they are adaptations to particular conditions that have arisen multiple times within the group. And they don't tell us anything about the original state of demosponges because they've modified themselves so far away from it that there's not much left there that's anything similar to what they started out as. What they started out as then was quite simple. Some sort of fibre network skeleton with monaxon spicules. And that gives us the two main branches of demosponge which we can trace back to, on the one hand, a fibre network skeleton of spongin and or chitin, and on the other a fibre network skeleton, mostly of spongin but sometimes chitin as well now, which has lots and lots of spicules embedded in it. So there are spicules on both sides and there are fibres on both sides. But the spicules on the keratosan side are very different to what we see generally elsewhere and are probably independently evolved. So unfortunately, this leaves us at a bit of an impasse. From the topology, from the molecular work itself, all we can say is that it could be either of these groups is a better model for the ancestral demosponge. You simply can't draw out the answer from what we can see from the modern ones. I mean, we know that there was a fibre network skeleton made of spongin and/or chitin, but we don't know whether that skeleton was embedded with lots and lots of spicules. We don't know whether spicules evolved in the Heteroscleromorpha and then independently within the Chondrocida, or whether spicules predate the Heteroscleromorpha potentially by some large margin. Anyone who's watched some of my other videos will know partly where that is going, but that's the question that is still left when you just look at the modern biology. We don't know when these monaxon spicules evolved within the history of demosponges. I guess that means we're going to have to go and find some fossils, isn't it? It's a hard life. The fossil record of demosponges is extremely mixed. So on the one hand you have things like the hypercalcified groups and the lithistids, which just, well, they make reefs, you know. They're not going to avoid being fossilised very easily. And we have an extremely good record for many of those groups. As long as the environments they were living in are preserved, probably you're going to get the sponges. However, they are a very small part of demosponge diversity. And you can imagine a lot of the rest are much less easily fossilised. So the keratosal ones, things like these organic fibre skeletons, these rot away. There's actually a big debate at the moment about whether they always rotted away or whether sometimes we do get them fossilised. That's something I'll come on to in another video at some point because it's fairly controversial. But we have not many examples of these that are widely accepted, shall we say. When it comes to the spiculate ones, things like these that wash up on the coastline, millions of tiny, tiny little needles inside it, bound together by soft tissue. They're living in a very aerobic, very turbulent environment for the most part, and they just vanish. These things disintegrate. They disappear almost instantly. So fossils of groups like that are extremely scarce. That's not to say that they're non-existent. I have one here from the Silurian, which you probably can't see, but it's a mass of little red branches, and this is yet to be described. But it's a very rare example of a sponge which has a spicule skeleton, a demosponge, which was living in a shallow water environment and has actually been fossilised. Mostly, they just disintegrate. After they've disintegrated, though, what about those bits of the skeleton? What about the individual spicules? Are they fossilisable? Well, yes, they are. You need unusual conditions not to dissolve them very rapidly anyway, but they can be fossilised, and you can, for example, dissolve them out of limestones and get quite nice material, especially in younger rocks. However, I need to introduce a new concept here, because everything we've talked about so far with regarding to spicules is the megascleres. These are the relatively large spicules that actually make up the bulk of the skeleton. However, demosponges and hexactinellids, but in a very separate way, also have microscleres. These are embedded in the soft tissues, and they have much more distinctive morphologies, and they're even tinier. Many of these you're looking at between 20 and 50 microns long, so very, very small things, extremely easy to dissolve in any warm water. So if in warm seas they vanish almost instantly, and they have an extremely poor fossil record. But if you do find them, then because they're such diverse things, they form all sorts of weird shapes like sigmas and forceps and toxas and chelars, and there's quite a lot of diversity in the form of microscleres. And if you were to look for those, then you might actually be able to get an answer to what was around at a particular time. Unfortunately, though, they just give us isolated anecdotes. We can say, for example, from a Cretaceous fauna that a particular family was around then, but they don't really give us that much more information. We do have some of the more generic types, like sigmas, going back to the Ordovician at least, where you have a type of microsclere that is fairly widespread within the Heteroscleromorpha. So again, it tells us, well, it gives us little glimpses into the past of these diverse groups of living sponges, but for most of the fossil record, we don't see a huge amount of them. It's just the odd, tiny little inkling as to what was there. Luckily, things get a lot more interesting in the Cambrian, and once again, it's the Burgess Shale-type faunas, mainly, that are coming to our rescue. Let's start with the organic skeletons. So, in the Cambrian, we have a family called the Voxiidae. These are known only, well, originally were known only from the Burgess Shale, but then they started turning up in China, in the Kylie and Xinjiang biotas, and they've also appeared in the Silurian in Scotland. So you have a pretty small pool of material to work from, but they now seem to be quite widespread even in the Cambrian. And they have a very simple, often two-layered wall of organic fibres. This is really interesting in itself, but some of those fibres have been shown to still be chitinous, which is absolutely extraordinary. And let's say that there has been some skepticism on this because it just doesn't seem possible. But basically every possible test was thrown at these things to try to confirm whether they were original chitin within these sponge fibres or some sort of modern contamination, including looking at things like radiocarbon, and found that it was isotopically dead. So this is beyond, at least it's older than the age at which carbon-14 disappears. So that's a good 100,000 years or so. It's at least older than that, and it appears to be the fibres of this sponge in the rock are still preserved partly as chitin, which is interesting. So the basic structure of this sponge, of Voxia in general, is a very semi-regular hexagonal network, mostly elongated, so you have chains of cells going up the sponge and then sort of slightly offset to the side. And it's a very simple form of what we see in the modern Varungids, for example. And it gets better than that, because the Xinjiang ones, the Chinese sponges, are also partly, it seems, silicified. The fibres have a silica signal when other fossils in the same beds don't. So it looks like they were originally partly silicified organic fibres. And that's really rather nice because, of course, it ties in quite neatly with what we see in the Phalangida. But it goes a little bit further than that as well, because there's one species in the Burgess Shale called Voxia bellula, in which there appear to be little monaxon spicules embedded within the fibres. Now this again has not been widely accepted, but personally I think it's fairly convincing, which is why I published it. And ideally it needs some electron microscopy, going back, getting some better specimens and just confirming whether they really are these little spicular elements in the fibres. I think they're pretty compelling. It's the same preservation as you'd expect to see in spicules. It's slightly distinct morphologically from the fibres, and it looks like you genuinely have a combination of this fibre skeleton in Voxia, including some monaxon spicules. This is potentially really important, of course, because it would strongly suggest that the keratosan and Varungid sponges, which today don't have spicules apart from those chondrosid little spherical ones, started out with a skeleton in which there were little spicules as well as the organic fibres. It's not all we've got to answer that question though, luckily, because we actually have a lot more information about what the sponges around the last common ancestor of the Demosponges were actually like. The other major group of demosponges in the Burgess Shale-type faunas are the Hazeliids. This is a family named after the genus Hazelia, which is actually quite abundant and very diverse within the Burgess Shale, but is barely recorded outside it. There are a few scattered examples. And what they have are relatively simple, conical, bowl-shaped to vase-shaped skeletons, occasionally branching, mostly just solitary, single osculum at the top, and a very thin wall, which is composed of an anastomosing network of little tufts of spicules, probably with organic fibres, but it's difficult to distinguish the fibres from the spicules in this type of preservation. The taxonomy of hazeliids at species level is an absolute nightmare, which tells you that they're definitely demosponges, which is, I guess, a good thing, isn't it? But mostly they have a fairly simple sort of skeleton which is parallel to the wall, just bundles and tracts of spicules. Sometimes they're more complex, they actually form little tufts which stick out from the end, or even tufts sticking out from the surface. So you have a wall like that and little spicule tufts sort of growing outwards all the way up it. So there are a range of structures within it, but they have a fairly basic form in comparison with anything that we see in the modern heteroscleromorphs. But it looks like a very simple heteroscleromorph. It's not quite like the living demosponges in arrangement, but it's not that far off, and it certainly provides a plausible basis for it. And these have, since they were described, been considered to be very, very early demosponges. None of them have any microscleres in. That may be partly because the preservation doesn't show them clearly, but it's unlikely to be that, because we do have sponges of this age which do show microscleres. Kind of. The first demosponge with microscleres in actually isn't even articulated. It's a disarticulated mass of spicules from the Sirius Passet biota. This is a Burgess Shale-type fauna, early Cambrian, North Greenland. I've not been there myself, but I got to work on some of the material. It includes this sort of mass of disarticulating spicules, which are simple monaxons, as you would expect in an early demosponge, but also it has C-shaped things, and then ones with a sort of fold in the middle. And these C-shaped things are called sigmas. It's a morphotype of microsclere that is very widely found across modern demosponges. Likewise, one with a bend in the middle, a forceps. We actually saw several types of microsclere morphology, but they were the same size as the megascleres. Exactly the same size as the ordinary monaxon spicules. So it looks like we're seeing an origin there of microsclere morphologies, which are just modified megascleres. They haven't reduced in size to become a different class of spicule yet. That being the case, it has to be a crown group demosponge, but also a stem group heteroscleromorph. So it fits quite neatly into a relatively small part of the tree. That's one of the bits we really wanted to see. We can see what the ancestors of modern heteroscleromorphs must have looked like. So this got a lot more interesting with the discovery by some colleagues of mine in Russia of demosponges which are rather similar from the Sinsk biota. This is also a Burgess Shale-type flora, but it's a little different because it's in much shallower water and has a very different fauna and flora, lots of algae as well, to most of these deposits. So we're seeing here a range of sponges, and it includes a paper, we ended up writing up a paper with 3 of them showing the same sort of structure. Is it simple body form, just a sort of vasiform structure with monaxon spicules as the main part of the skeleton, but also sigmas. And in one of these, the sigmas are the same size as the simple monaxons, so much as we saw in the Sirius Passet sponge. But in others, these spicules are much reduced. They're down to the same scale you would expect for microscleres. So we're actually seeing a sequence of fossils, which are not identical to each other otherwise, but they're very similar, which show the change from megasclere size sigmas down to very typical microscleres, as you would expect to see in modern demosponges. So now we are seeing stem group keratosans plus pharongiomorphs, which appear to have monaxon spicules embedded within the fibres, and we are seeing stem group heteroscleromorphs, which have the origin of the microscleres. And all of them seem to come back towards something which looks like a Hexactinellid, something with a basically simple wall-parallel array of spicules in little anastomosing tufts and bundles, probably embedded within organic fibres as well. The Hexactinellids are now widely accepted to be very close to the basal group of demosponges, and quite likely actually contain the origins of these two major branches of demosponging. Can we take it back any further? Well, yes, and the next stage is a thing called Chromulospongia. This is again a Burgess Shale one, but has turned up in a few other places, at least tentatively, around the world in the Cambrian. And this one has the same basic structure, but even simpler. It is simply a single wall of spicules, all wall parallel, there's no tuft sticking out. It's forming a sort of reticulate anastomosing network like in Hazelia, but the body form is much more consistent. It's a sphere, a globe, with a hole at the top. It's a very simple structure which is very similar in form to things like the reticulosans, which have normally been assigned to hexactinellids. And the wall also is pierced by an array of holes, sort of large ones surrounded by small ones, quite a irregular pattern. It doesn't show any of the chaotic irregularity, the plasticity that we know and love from modern demosponges, or even from Hexactinellids. This is a much more constrained form, but with the same structure, the same basic fundamental structure of how the spicules are arranged and what sort of spicule they are. They're all these tiny little monaxons. So this is also generally accepted to be a very early demosponge, and based on everything we've seen so far, it's most likely in the late stem group, leading up to the Hexactinellids, where you get this big explosion of morphology and all the diversification going on then. So what about even further back than that? For this one, I'm going to take a great leap backwards, in many senses, to one of my first papers. In fact, my first paper on sponges, second technically in the same year, which was absolutely dire. This was going back to 2003, when I was just working out how to deal with sponges and nobody was telling me. I was working it out on my own, and therefore I was putting things in print that I probably really shouldn't have done. But one of the things that I came up with was looking at a thing called Cyathophycus. This is a sponge which has actually become quite important in later years. It has a regular square grid of hexactins. The shape of the sponge, though, is just like Chromulospongia. It's a sort of a sphere with a top chopped off, with a hole at the top for the osculum, but it's basically a nearly spherical sponge. But a regular square grid of hexactins covering the surface. But that's only one spicule layer. And in some of the Cyathophycus species, especially, you have an inner layer, which is a dense thatch of tiny little monaxons. And for some reason it struck me that if you just sort of lose the hexactins, then you end up with a ball of a thatch of tiny little monaxons, and it doesn't half look like Chromulospongia or Hazelia. And this is the sort of thing that really probably shouldn't get published at all, but it turns out I was probably right, by pure fluke. So the first stage in this part of the argument was back in 2013, when we started looking at some new material of the same species of Cyathophycus in an electron microscope, and found that the preservation of the spicules was absolutely exquisite, down to the internal microstructure, multiple layers of mineral and organic matter, which is unusual in itself, but main thing for the purposes of this video is it preserved the axial filaments, or the axial canals, and their symmetry. So when you have a spicule secreted within the demosponges or hexactinellids, it is secreted onto an organic filament, a tiny thing about 5 microns wide usually, which is basically a rod of enzymes that other cells then secrete silica on top of. It acts as a template and an inducement to crystallize. So this is quite an interesting thing, because in the hexactinellids, this axial filament is square, absolutely universally. It is always square. and this appears to be related to the packing structure of the enzymes that are used to secrete the spicules. In demosponges, they're never square, they are triangular or hexagonal. Same symmetry. And again, slightly different enzymes being used for the biosolidification, and so you have a different packing structure and you end up with a different symmetry of the axial filament. So we could see the symmetry of the axial filaments in Cyathophicus, and it turns out they were hexagonal. This should not be, because of course that is diagnostic, as I've just said, of demosponges. It's not a hexactinellid axial filament, it's a demosponge one. So this, at least taken at face value, seems to be showing that this thing, which has a regular grid of hexactins on the outside, but an inner wall which looks remarkably like an early demosponge, has a secretion process that belongs to demosponges. Which means that, very likely, this thing is actually a hexactinellid-like ancestor of demosponges. This is controversial. It's still controversial. Even now there are some people, actually there are various schools of thought that suggest that we cannot possibly have spicules in the early demosponges. This seems to be a widespread assumption, and therefore this must be wrong. So, of course, what you really want to see is something intermediate, something which is clearly demosponge in all sorts of ways, but still retains hexactins. That would be the ideal. And that is where the angibiota comes in. The angibiota is actually latest Ordovician. It's quite a bit later than the Cambrian Shale-type faunas, but of course things can survive, lineages can carry on for a while before they go extinct. And there's a whole video about this, introducing it, if you'd like to go and read up on this one. But it's an extraordinary deep water post-extinction sponge fauna from South China. And we first described it in 2017, and in the same year we also published what we thought was one of the most extraordinary sponges in that deposit. We called it Conciliospongia angiensis. And this one, we actually included in the title the idea that this was a missing link between hexactinellids and demosponges. A little bit clickbaity for an academic paper, but we thought it justified it, and still do, even more so now. What we have here is another globose sponge with a body wall which is a little thicker than Heseliids. It's actually got a certain thickness to it, and you can see it's made of an array of tiny monaxons in a typical Heseliid-like structure, but with tufts of those spicules coming outwards to form columns almost surrounding internal canals. So it's actually a slightly more complex structure than any of the known Heseliids. But it is based on the same arrangement. So you've got the same wall shape, you've got holes within it, and you've got an array of monaxons which then are being extended outwards to form these sort of columns of little spicules. And that's all fine and good. It's a perfectly good demosponge. And it's got hexactins in. Not huge numbers of them, and they're mostly concentrated around the upper part of the sponge. They're all consistent morphology, they're all complete as far as we can see, they're not broken fragments or anything like that that have been washed in. It is quite a rare sponge, but it consistently has this structure which includes a reasonable number of hexactins embedded within this skeleton of monaxons. So it's basically got a demosponge skeleton, and it has little hexactins in it. Exactly as you might hope if it was part of a lineage that had primitively started out with hexactins as well and had gradually lost them. Because the skeletal wall with organic fibres and lots of tiny monaxons in is far more flexible in evolutionary terms. You can do much more with it. And it gives you the option of becoming morphologically plastic, of adapting to all sorts of environments, as the demosponges subsequently have. Now we are working on Conciliospongia again. We have an extra specimen which we can do more with, which preserves extra fine detail. And because we're writing up the paper and it's with colleagues in China, I can't tell you exactly what, but let's just say that the story is being reinforced. it really does seem to be a demosponge with hexactins in it, which is rather neat. When you combine all this together, what you get is a really lovely potential evolutionary sequence from a hexactinellid-like reticulosum with demosponge-like axial filaments through to a purely demosponge-like skeleton which has a few relict hexactins in, to these very constrained bowl-shaped sponges which then become more flexible in their body form around the base of the crown group of demosponges. And from there we see this very rapid diversification into the keratosans and phalangids, and also the heteroscleromorphs, where we start to see the origin of microscleres from megascleres. And it's a really lovely story. The only thing missing, you could say, is that we don't have many of the intervening stages in the critical, very early part, in the stem group. We have quite a few, and they seem to show us exactly what I would like to see. But it would be nicer if we had more of them. But even so, from what we can see, either there's a lot of convergence going on somewhere, or we actually have a genuine evolutionary sequence that makes perfect sense. It fits in perfectly with the topology that we get from molecular phylogenetics. So we know what shape the evolution sort of takes. It matches up with the distribution of characters that we have in the different groups, and it just gives us more of the backstory and shows that they started out as these more constrained sort of globose sort of forms, which apparently started out as something that looks like a hexactinellid. That's where it gets really controversial, because a lot of people argue this cannot really be the case. The reason for that is if you have a stem lineage of demosponges which have spicules, and the base of the crown group of demosponges had spicules, then we should be seeing fossils of sponges going a long, long, long way back. So, what is the oldest known demosponge? Probably, if you're going to be conservative, it's Vauxia leoia from the Chengjiang biota, which is early Cambrian. Between there and the Burgess Shale, almost all of the critical species have turned up in that time interval. So it's a relatively short window where we're seeing the earliest definite demosponges, and then most of the range of diversity going into the base of the crown group. There are also isolated spicules, which go back further, which may be demosponge, little monaxons, for example. And these even go back to the base of the Cambrian and even just into the top of the Ediacaran. The problem is that it's not only demosponges that make little monaxons. In fact, you can find them in every single class of living sponges. We don't know what the original mineralogy of these things was, and we can't say what these things were. I mean, some of them look vaguely like some particular type of demosponge, which is a derived form in the modern, but they just explored every possible variation of little monaxons, so that doesn't really mean anything. And there's probably quite a lot of extinct groups which also secreted monaxons, so we can't really do much with that. There's another candidate, though, for the earliest one, which is archaeocyathans. And these are a group of still somewhat contentious organisms that look like they should be early demosponges and have been argued to be as such. Based on everything from the morphology, the structure is vaguely similar to what you see in the early Vauxiids, for example, but they have a hypercalcified skeleton with no spicules, no organic fibres, and so on. And so it's very difficult to be certain that's what they are. However, there was one study on the immune responses that are inferred by the reactions of these sponges growing next to each other, which implies a relatively complex immune system as you get in demosponges, but not hexactinellids. So it is quite plausible that these were actually early demosponges. And if so, then the archaeocyathans push the origin back to somewhere around 10 million years before Chengjiang, so still well above the base of the Cambrian, about 8 million years above it, and still in a relatively short window of time. This is actually a huge problem, because molecular clocks have the demosponges appearing around normally 600 to 700 million years ago, and that is the crown group. So the crown group are the things that we can do something with. We see them abundantly in the Burgess Shale. You get the lithistids starting to form around there as well. They are, the one group, the ancestry, is an early branch from probably the Voxiids, something like Voxia bellula, Feldospongia bellilineata, and you end up with a sequence leading directly to the first lithistids, and possibly archaeocyaths as well. They're very early on in demosponge evolution within the crown group. So we have this problem that we expect to see demosponges in some form based on molecular clocks going back 600–700 million years. And that's just demosponges. If the spicules are shared with hexactinellids, then that pushes it back even further, much, much further. Maybe 700–800 million. It becomes a little bit ludicrous when you're dealing with things that actually have spicules that are preservable, especially in the Precambrian when dissolved silica levels in seawater were very high, and so it would help to preserve spicules rather than dissolve them. Another line of evidence that is often used is demosponge-specific biomarkers, which go back to, again, about 700 million years. These are things like 24-isopropylcholestane. They're basically steranes, which are modifications of sterols, which are a group of compounds that are very widely distributed among life in general, eukaryotes. Possibly even further than that, but certainly eukaryotes. And some of them have very distinctive sort of forms or structures, and you can potentially get chemical fossils, where you can extract the sterols from an ancient rock, analyse them, and look at the distribution of sterols through life as we know it, and say, well, this only occurs in that group, and therefore it's one of them. And this has been done, this is actually quite a long-running saga, for various things that have been assigned to demosponges. The problem is that in more recent work, it's been shown that as you sample more things like rhizarian protists, they also produce small amounts, at least, of the same sterols. And even worse is that what we're seeing is after geology has had a go at it. So some sterols actually get converted through time to mimic others. And it turns out that one of the ones, the critical ones for interpreting the origin of demosponges based on these things, is just as likely, if not more so, far more likely in fact, to have been derived from algae and then modified. So we now know that these are not reliable sources of information. There are still more papers coming out suggesting more demosponge-specific sterols being found, but the problem is you're looking at one character. The sterols are a diverse group. We don't know that much about which groups have evolved different sterols. It's certainly not impossible to have convergent evolution of a methyl group on the end of a sterol, and we can't use that on its own, because everything that we know about it so far has been undermined. There was also, a few years back in Nature, a claim for a possible crown group, well, Keratosan demosponge from rocks 890 million years old in Canada in a single thin section. And this has been part of this saga about, can we identify things like this in limestones? And the answer seems to be probably yes, but very, very carefully. Because when you take a cross-section through that, what you see is a little network pattern, effectively, or a series of dots, more likely, because you're cutting through fibres rather than through a honeycomb. So there are many different ways of creating textures like that, from the spaces around a clotted fabric of little pellets, microbial aggregates, even alteration effects. And there's now been a lot of work, which was actually stimulated by this paper by Turner, to try to really get to grips with what are we looking at here. And the verdict is that in most cases, probably there are alternative explanations. There are some that from the Paleozoic, which probably are demosponges, I think. But most of them, where you just sort of take a little network thing in a thing section and say it's a demosponge, probably not. And this one from 890 million, nah, it just isn't. So where does this all leave us? That's a really good question. But rather like with the other groups of sponges, we have a bit of a mystery here, a bit of a problem, because we actually have a very good and useful fossil record in the Cambrian. It's very, very patchy after that. But in the Cambrian, we have what looks like a nice coherent story about the diversification of the crown group of demosponges, and even being able to trace that crown group back into the stem group. And there we seem to see continuity with the Hexactinellid-like reticulosans. So I find the evidence for this history really quite compelling. But even if we don't accept all of those lines of evidence, or all of the links in the chain, even if somehow there's a way of getting around some of it, there are still certain things that we can say with very high confidence now. So one thing is that we know what the body form of the earliest demosponges was like. It was not like one of these, or one of these. It was not compound, it was a simple globose to vasiform shaped thing with a thin wall. It hadn't developed all of the complexity that we see in the colonial encrusting forms that we get today. Most of what we see in the diversity of modern demosponges, these amorphous blobby things that have been the inspiration for these ideas of them being basically colonial protists, that's actually all wrong. That's secondarily derived. The ancestral forms were much, much simpler in terms of their architecture, and therefore much more constrained genetically. They're not blobs. They are actually quite well constrained, vase-shaped sponges which look remarkably similar in form and structure to what we see in the other classes of sponges at the same time. They all go back to the same sort of thing. So all of these derived demosponges that we see today are derived. It's not the primitive state. So that's one thing which is actually really important for understanding where sponges came from. They came from something with axial symmetry, a thin wall, large central chamber. There you go. And then you have to say, if demosponges appeared even in the crown group form 100 million years before we see any of them, why do we not see any in the Ediacaran? That's one question. Okay, you might start saying, well, it was very weird preservation, they were living somewhere else, all that sort of stuff, but it doesn't get around the wide range of preservation that we have. But that's only one part of the problem, because aside from that, why do we see this divergence of the base of the crown group in the Cambrian? Why does everything that we see that looks like a demosponge in the Cambrian look like it was around the base of the crown group? We actually see this divergence, the voxiids, the hazeliids, these ones with the sigmas in them. What's going on there? If they actually evolved something like 100 million years earlier than that, why have they all stuck around invisibly for 100 million years into the Cambrian, and then they suddenly appear, and yet we haven't seen much more derived demosponges turning up with them? What's happened in that 100 million years? It looks like someone put them into the deep freezer and then just sort of thawed them out 100 million years later when we want them. It doesn't actually make much sense. It's one of these cases where you have to start to really get to appreciate what the fossil record is telling us. Certainly what it can't tell us or what it doesn't tell us, because it's very incomplete. The fossil record is by no means perfect. We all understand that. But when you have enough information which all falls into one consistent story, why would you disbelieve it? There's a contradiction here between expectations from molecular clocks and the biomarkers, and predictions, and the reality of what we actually see in the fossil record. And I'm a paleontologist. I find genetics and molecular clocks and all the rest of it are extremely interesting and useful tools and models. But when it comes to actually seeing what happened in the history of life, for me, the fossil record is the way to go. So I will leave it there. There's a bit of a conflict. There's a lot we still don't understand properly. But the story that we do seem to be seeing seems to be that these lovely things actually have quite a well understood history. And yeah, there's not much we can do about it really, apart from go and find some more, see if we can fill in the gaps, see if what we find contradicts these ideas or gives us more dimensions to it. But I will leave it there because it's already been quite a long one. But yeah, I'll have to work out what the next instalment in sponge history is, but I'm sure it's going to be really, really exciting, isn't it?